Great. Good morning, everybody. So we've got a very exciting program today, and it's my great pleasure to introduce the first speaker, who's Dr. Manel Juan. He's Head of Immunology at the Hospital Clínic in Barcelona, and he's also a research leader at the ECDGA, which has a very long Spanish name that I'm going to just abbreviate to ECDGA. And he's been instrumental in the implementation of European CAR-T therapy, and he's going to tell us today some of the recent advances connected with that and potentially new applications. Thank you, Dr.
Thank you very much for your kind introduction. I want to start thanking also to the organizer for inviting me to be here, specifically to Dr. Solanik, that we have a good relationship for a long time due to the fact because I am immunologist and both are involved in the management and trying to give the best diagnostic for immunodeficiency. But now, today, we will try to explain things about a big opportunity that we have, that it's the use of cell immunotherapies to treat different diseases. I will try to convince you that this is an opportunity for everybody, specifically for rare diseases, specifically maybe also for this ECD disease. So this is my disclosure that can be summarized in the fact that I don't have any economic profit for anything. So at the personal level, I was working in a public hospital and I manage all my elements at the academic level. It's interesting to start in one thing that you obviously knows perfectly. It's what is this Chester disease, this ECD disease. It's clearly a rare clonal expansion of histiocytes. I take this for your web page. And I think it's interesting to have in mind that even there are too many different expressions that you know better than me, there is specific changes at the level of somatic mutation for several genes. Being BRAF, probably this mutation, they were more clearly identified in most of the cases. I want to specifically point out this point because I think it's important to have in mind what we try to do. Immunotherapy has arrived to the cancer area after change a way to see the treatment of cancer. Until now, we have the tumor in the body and we use external elements like surgery, chemotherapies, and even also specific molecules for changing specific targeting changes. And all the treatment are focused in the tumor, are trying to look at the cells and kill the cells or eliminate the cells, but any way you have in mind. Immunotherapy is based in the immune system that frequently, or in most of the cases, we don't see the tumor because it works very well. Immune system is able to recognize tumors. And in most of the cases, it has been calculated even that between 5 and 10 years will be enough time for more of the cases for develop a cancer. And unfortunately, we don't have each 10 years a cancer. And one of the reasons is the fact that the immune system is able to recognize the change and eliminate the tumor. And what we try to do in the immunotherapy is to increase this function of the immune system. We already have this function of the immune system. It's internal, and we want to improve what we have inside. Can be continuously effective. All the others that come outside, you make one specific intervention, but after the intervention, this function doesn't continue in there. When you change the immune system, you already have it, and you know perfectly because vaccination in infections, it's clearly one of these long-term working of the immune system, and it can be extremely specific, targeting specific changes in the body. And this probably is a big change in the way that we treat cancer, at least for cancer, but probably for other things. Immunotherapy is one way to change the evolution of different diseases. I love to use this movie of the Alex Ritter lab, where you have here a lymphocyte and a big tumor cells, because this summarizes what we are trying to do. We are trying to increase the function of these lymphocytes, being able to eliminate the tumor. This is the purpose of the immunotherapy. Sorry. And by now, after the success, because we need to know that for cancer, surgery, chemotherapy, and radiotherapy are a very successful treatment, now we have other options based in the use of monoclonal antibody, but also in the use of cells, because finally immune system is defined by the cells. And we can go directly to the cells to try to obtain the function, in this case, kill the tumor cells. And the first way to do it was developed in Seattle, in Fred Hutchinson Institution, where allogenic transplantation provides a way to eliminate tumors through allorecognition. But this is the oldest way to do immunotherapy, based in allorecognition, but along the last 30 years, it has been appearing a lot of different treatment that has been able to arrive to be used for this immunotherapy treatment. I will talk mainly in the right side of this slide, that it's CAR T cells and also the use of transgenic TCRs that has been demonstrated that it's possible to treat. One of the big changes in relation to the use of stem cell transplantation is that all these approaches has been considered drugs. And this has been a change in the way to manage this kind of treatment. Transplantation, stem cell transplantation is a procedure, and the rules of use this treatment is based in the rules of the procedure, like other big numbers of procedures in the medical treatments. But when we talk about this cell immunotherapy, we are talking about knowledge, quality rules, infrastructure, clean rooms specifically, that make this a little more complex. In any way, I will try to use this. I have another video at this moment in the morning. I think this can be a nice way to start, where you have here a lymphocyte and too many different tumor cells. And you can see here how this cell is contacted with one, with other, another, and you can arrive to kill even 1,000 or 10,000 cells by just one lymphocyte. This is an incredible number or capacity of the immune system to obtain this capacity to eliminate this option. If we use this image in a cartoon, the green cell is the lymphocyte, this is the tumor cells. There is at least 3 major molecules or 3 major levels of molecules that make the change in the lymphocytes to be able to activated and make the action against the tumor. These 3 elements can be summarized in the capacity of recognition, the specificity that has been able to develop due to the fact that there are very specific antigen receptors, in this case TCR, because we are talking about T cells. This is the major cytotoxic T lymphocytes. There are also NK, but this has been another approach. But in general, we are focused in T cells. And you have the first signal based in T cells due to the signaling of the TCR, and CD3 is the molecule associated in the signaling of the T cells. But also there are other molecules, CD20A mainly, that make second signal and it produce proliferation and cytokine production by these cells. But for cancer, and specifically for any kind of treatment that you should or you want to have this option of recognition for a long time, and human, it's different for experimental models in mice where the life is shorter. But here you want to have this for a long time. Survival signals are very important. And here there are too many different molecules, and 4-1BB is probably one of these interesting molecules. In any way, how we can change the lymphocytes to direct these lymphocytes against the target, in this case the tumor cells or whatever was there. the first element, the more important element, is the first signal, the cytotoxic or the activation signal based in the TCR. So if we introduce a TCR that recognizes the target, you can arrive to have the capacity to modify your lymphocytes and focus your lymphocytes against the target. This is the first approach. The problem is that this recognition of the TCR is based in the capacity to recognize the difference, the antigen, inside the HLA molecules. And we have too many different HLA molecules. So we need to develop a lot of TCRs, not only for one specific antigen, that can be a lot of different antigens, but also for each HLA molecules. And we have several in the body. So this approach has been already demonstrated efficacy, because we have developed many different clinical trials. You have here a list for different antigens, tumor antigens, and you can see here that there has been different approaches against different HLA, in general HLA-2, because it's the more frequent allele that we have. Based in this concept, you are able to introduce a TCR that allows that your lymphocytes has arrived to be modified to focus against the target. This has been successful and has been approved by FDA for a specific treatment based in a specific TCR against a specific antigen tumor for HLA-A2. This has been the way to demonstrate that this is possible, but in any way it's quite difficult to define all the TCR for all the different antigens for all the different HLA. And this is the reason, even there are TCRs against this major mutation in ECD that has been defined by the Riedel group, also in Seattle, by others, and in other articles you can see here. And even there are patents based in the use of this, not for ACD, but also for other tumors that has this mutation. This can be an option. It should be arrived to be an option for the treatment of specific or with highly specificity against this variation, this mutation in the tumors. In any way, this is very complex, as I tried to demonstrate, and this is the reason why appears a new receptor that are not mediated or are not defined by the presence of HLA. And the receptor that is able to recognize antigens directly without HLA is the antibody. The problem is that if we put an antibody in a T cells, the T cells doesn't work. And this is the reason that along the last near 30 years, 20, a lot years, has been developed that we name chimeric antigen receptor. That is just the combination. It's a chimera that we produce molecularly, combining the recognition domain of antibodies with the signaling domain of the molecules that are involved in the activation of the T cells. And this is what we name chimeric antigen receptor, or CAR. It's a combination of the different things, and this is the reason why we use the name chimeric. Has been developed more than 30 years now by the different investigators. The evolution of this CAR arrived for the use of the different signaling domains. I explained that there is one first signaling, a second signal, third signal that are important in cytotoxic proliferation and also survival of the cells. And this is how has been developing in the last time CARs, in the first generation CARs based in CD3, that is the signaling of the TCR, that induce cytotoxicity, and the cells kill very well with this approach, but we need to have an improvement. By this way, the cell killed, but finally the T cell also dies. So to proliferate and to introduce cytokines and arrive to have better option, we develop second generation CARs with different molecules combined in the signaling area being CD28, the more clear one, but having other options like CD137, that is 41BB, that has been also able to demonstrate to introduce survival signal. These are the three more clear generation of CAR T cells, but by now, the CAR that we have been using now and has been approved for being used in the treatment of different hematological cancer are this one, second generation CAR. It's important to have in mind that when we introduce a gene that codifies for this CAR, we are introducing this in a T cell, in your own T cell, that it's a T cell that exists for other purpose, not for killing your tumor cells. So they have already their own TCR. They can be against a virus, they can be against anything. But this TCR, the capacity of recognition of this TCR, it's changed by the capacity of recognition of this chimeric antigen receptor, recognizing a tumor antigen in the tumor cells and developing their function. If this T cells is a cytotoxic T cells, these T cells with a CAR that we name CAR T cells is able to kill by cytotoxicity to kill the tumor cell. But if these cells is a regulatory T cells that it's inhibitory, we will have an inhibition of the function of the immune system. And if you put this in a specific helper T cells, you introduce a specific cytokine in the capacity of the function. This approach is very interesting because even the CAR T has been considered a drug. The product, the T cells, the modified T cells has been considered a drug. In fact, by now, all the approved CAR T cells are autologous CAR T cells. And this is really a procedure where you take a patient, you decide if this patient is able to be included for the disease. And we obtain their own cells by leukapheresis of the patient. And after changing these cells by introducing the gene by any kind of ways, being viral vector the more frequent way, there has been this viral vector, it's a vector that has the construct of the CAR T cells, we arrive to have by transduction the T cells modified with this gene. And after grow these cells in a bioreactor, we obtain the product. This is what we name a drug, a CAR T cell drug, and what we use for the treatment. But we use this treatment just in the same patient. By now, we are trying to make changes and be able to arrive to have for a donor and to have allogenic CAR T cell. By now, all the products are autologous. And in this autologous procedure, we have two major steps. It's very important to make in the best way the product, but also it's very important how do you manage the patient and how do you modify the situation in the patient. I will try to convince this. This is finally, even it's considered a drug, and drug agencies are regulating this authorization of use. Finally, it's more similar, and I'm absolutely convinced, and even everybody said no, but I'm absolutely convinced that it's more similar to a procedure like transplantation. In any way, this is the situation that we have with a lot of clinical trials in the world. We have arrived to more than 1,200 clinical trials in the world, with China and States in major areas, in Europe very small numbers of clinical trials, but in Spain specifically it's not a bad place, and we have been able to demonstrate this. Probably the first success in the treatment, there was others, but this was done in August 2011 with this starting article, New England Journal of Medicine, under the direction of Dr. Kaljun in University of Pennsylvania. And this is one of the 3 patients treated in this moment that was treated in 2010 and arrived at 2020, 10 years later, free of diseases. It's important to say that this treatment has been developing in patients without other options of treatment. So in these patients without other option of treatment, we obtain good results, enough to at least to be 10 years free of disease. As you can see here in 2020, and you obviously remember this moment, everybody wear masks because it's a pandemic moment. And unfortunately, 1 year later, the patients die, not by the cancer, die by COVID infection. In any way, probably the more clear image of the success of the treatment with CAR T cells arrived for this girl. That was the front page in one of our more important journals in Spain, but also in the States. It's Emily Whitehead. It's a girl with leukemia, and acute lymphoblastic leukemia, that has been able to survive also 12 years. And probably the older treatment arrived for the second patient in this seminal article of 2011. It's Doug Olson. It's in fact a medical doctor that also received the treatment with CAR T cell and is still alive after more than 14 years now. So it's free of disease. It's interesting to know that this is not the first patient treated with success with CAR T cell. In February or January of this year, February it published, this article demonstrates a patient that after 18 years still survived with a neuroblastoma by the treatment of CAR T cells of first generation CAR. Other CARs are second generation CARs based in the concept that it's enough. But in this case, due to the fact that we use T cells specifically selected against one virus, Epstein-Barr virus, the TCR, it's by themselves able to signal inside the T cells and combine this signaling with just one first generation CAR, and even it's not a very good treatment for most of the patients. In this patient, 80 years later, it's enough to maintain the function against the neuroblastoma. You are able to detect T cells against this antigen, GD2, that is able to kill. In the other cases, you have a lot of pictures of the patients that survive for this, but in this case, the group doesn't want to present the face of this girl, 18 years now of survival, and just you have the hands here of the patient. It's interesting to note that we have a lot of CAR T cells already approved, mainly CAR19 for leukemias and lymphomas. These are the 4 for leukemias and lymphomas CD19 positive cells, and also for multiple myeloma against this BCMR. The problem is probably, at least in Spain, in other countries also, is the treatment for the drug. It's 300,000 euros per patient. So it's a very expensive treatment with this CAR19. In any way, probably after 8 years of treatment, where we have a good results of success of treatment by all the elements analyzed by the use of this, after 8 years of approval by FDA and 7 for EMA, this is the distribution of these CARs in Europe. Here you have the different countries, and here you have the different products. And in green is where you have access of this, and in red is where you don't have access of this treatment. And it's clear that this is a failure, because after a long time, there is a lot of patients with a capacity of being treated by a successful treatment like CAR T cells. There is a lot of way to improve CARs. You can introduce different molecules, and I will explain something. You can introduce different cells. It's not just T cells, it's also NK, also macrophages, safe treatment. Just one patient for more than 40,000 patients probably have genotoxicity. It's not clear, but it's the only case that where it's possible that the introduction of the gene in the T cells modify these T cells and develop a new tumor cells in the T cells. In any way, this is an option, but having in mind that we are treating with other options of treatment more than 40,000 patients. This safety allows us to have in mind the possibility to introduce new changes in the T cells. And probably this is, has been starting with this armored CAR, where next to the CAR T cells we introduce another gene, a cytokine, an antibody. We are able to produce T cells that produce antibodies, the antibody that we use. Even we are able to produce or to introduce genes for make the T cell produce a drug. And this drug can work inside your body due to the fact that your T cells are producing all these things. So the capacity of making this, and for cytokines we use the name drugs because it's a very clear way to understand. It's like a car that carries something else, in this case a cytokine, can be an antibody, can be everyone. Armored CAR, it's another option, and we are able to then find different generations that improve the function of this in different aspects, moving the cells in the correct way and making all this possible for different, not only antibodies, but also ligands, like the own cytokines or any kind of adhesion molecule in there that make possible to use in autoimmunity, in cancer, and even in transplantation, killing the cells, if we use cytotoxic cells for elimination. Sorry. We're here. Okay. And remember, we are able to make cells that secrete these antibodies or secrete drugs. And this has been defined for different authors here in Spain and also in the States. And this is another option that can be combining with different cell types, subtypes, current case, and having in mind the fact of this case that already explained, you are able to introduce this in a T cell that is able to recognize a virus, and by this way, a CAR of first generation is able to remain in the patient for more than 18 years. Remember that this is an autologous procedure, and we were able here in Barcelona to define a new capacity of production of this CAR T cell by our site. And we defined the two steps to produce the lentivirus, but specifically to make all the procedure to make the product here at the academic level in Barcelona. And here is the Hospital Clínic, and we have the pediatric hospital in this southwest area of the hospital, very near the Hospital which is more or less in this corner. And we are able to define here the capacity of produce in our hospital. This has this U shape. We have in one of the corner the capacity of make the viral product. In the other, we make in a very manual way, but now we have the capacity to scale up by bioreactors. But in the clean rooms, we are able to produce in this area by different bioreactor products that allows us to treat in this moment more than 500 patients after 7 years. And we obtain this product that has been authorized by the Spanish Drug Agency for being used in patients, CAR-19, based in a specific procedure with a clear, good results comparable to others and very small number of adverse effects here. You compare both graphs. This is our graph, and this is the graph of the best clinical trial in this indication, and it's very similar both. And we arrived to demonstrate that this it's good enough to be authorized by the Spanish Drug Agency, and now we are moving to the EMA. And by this way, an academic product without companies involved in this, we have the first authorization in the world, and we expect to use this for the future. We have a second one that has also the authorization for BCMA and allows us to use our product as a way, at least in Spain, to treat our patient in success. We have others, RE3, with different levels of capacity production and success in production, with different targets, not only hematological tumors, but also solid tumors, and outside this, also in autoimmunity and rejection. and we expect to be able to arrive to treat specifically solid tumors, and specifically very soon to start with glioblastoma in patients by using different approaches, and one specific approach based in targeting one of the antigen of this. Having in mind that autoimmunity has also a success capacity of treatment by the use of CAR-19, not only lupus, but also neurological and other diseases, like you can see here in autoimmune diseases. It's important to have in mind that we are just starting monotherapy, and probably nobody is trying to treat cancer with monotherapy, but we are treating cancer now with immunotherapy in just one product. And it's possible to combine all these products and change the capacity. And we are convinced that this general scheme where academy start all the hypotheses and later to transfer to pharma is able to remain in the academy. It's probably important for being able to use for patients, specifically when the number of patients are very low. With rare diseases, it's very difficult that companies decide to invest for being able to use this. Academic network is important. We have in Spain, in Europe, and also in the world. And we expect to combine these more than 300 professionals for different specialties to work together, and the sponsor that allows us to have the money, mainly non-profit sponsor, not only public institution, but also some structure like this foundation, bank foundation La Caixa, that next to this different networks allows us to treat all together hematologists, immunologists, the direction of the hospital, and a lot of people, and arrive to make this treatment. Thank to everybody. Obviously, thank to the patients that allows us to demonstrate this, and thank also to the sponsor. But also thank to regulatory agency in Spain that allows us to know the best way to arrive to have success in this treatment. The immunotherapy start in a very manual way. CARs start to put a different proposals with some problems, obviously. For big arrival to solid tumor, we need probably better CARs, stronger CARs, but also it's possible to arrive to the higher place by changing the microenvironment, and probably this is one of the areas. In any way, CARs has been multiplying and arrived to have options for the treatment, and probably we have started a new way to dream new options of treatment and arrive in this pathway later. So thank you very much, everybody, for your attention. This is our team, and this is the celebration of the 500 treatment last November. So thank you very much.
I think we have a couple of minutes for some questions.
Thank you. That was very exciting. So you talked about autologous CAR T cells. Are these off the shelf, or the new ones, the second or the third generation? Are these off the shelf CAR T cells?
All the off the shelf, all the allogenic products by now has been demonstrated to be less effective than autologous, because having in mind that the product is just T cells and the immune system doesn't work alone. Your T cells are very important. It's able to control everything, but your immune system, it's a key element. And the interaction between this product and your immune system is better if your own cells. When you use other cells, the things are not so good. And in general, it's an option to have the off-the-shelf production, but in general, until by now, and probably in the future this will be changed, but by now it's clear that it's better to use autologous procedures. NK cells can be a way to avoid this, but by now also has been not able to demonstrate this. And if you want to have memory, you want survival of T cells, NK cells are not good options. So T cells allows us, you to make this possible.
Okay. So thank you very much, Dr. Juan. Wonderful talk.